So good morning. I would like to start out by welcoming you all to this second lecture of the Omics lecture package. I will talk about proteomics and drug development. And I'm Magnus Jakobsson, an associate lecturer at the Department of Immunotechnology, focusing on mass spectrometry-based proteomics and its applications. So the lecture objectives, what you're expected to learn and account for after this lecture. You're expected to learn about the basic principles of mass spectrometry and its application areas. You're also expected to be able to describe one application of mass spectrometry-based drug development. And the examples I will talk about relate to drug target identification, characterization of drugs, and finally repurposing of drugs. And in the lecture I will start out by giving you an introduction to general omics and proteomics in particular. I will then move on and talk about the mass spectrometry, its basic principles, and how a mass spectrometer can be used to quantitatively assess differences in protein abundance between conditions. And I will then finally go into 3 examples where quantitative mass spectrometry is used for the discovery, characterization, and repurposing of drugs. And the examples will relate to plasma proteomics, drug-protein interactions, and protein-protein interaction networks. So first about the omics and proteomics. This lecture is part of an integrated omics lecture package where Ashfaq Ali previously talked about the integration of different omics dimensions. And the different omics can, for instance, be genomics, transcriptomics, proteomics, and metabolomics. And the conceptual idea is that the integrated analysis of these different omics dimensions allows you to do a systems level analysis and interpretation of health and disease states. And this can allow you to discover new drugs and drug targets, and also provide insights into pathogenesis of diseases. But my lecture will mainly relate to proteomics. So I would like to start out this lecture on proteomics by defining the proteome. There are many definitions out there for the proteome. One that I like is that it refers to the complete set of proteins confined in a biological entity. And this biological entity can then be defined on an organism, organ, cell, or organelle level, but it can also represent biological fluids. Looking into the human cellular proteome, so a typical human cell expresses roughly 12,000 proteins out of the 20,000 protein coding genes. And these collectively make up 8 billion protein molecules, and they have a dynamic concentration range of between 1 and 10 million, and a collective protein concentration of 300 milligrams per milliliter. So the proteome already at the cellular level is extremely complex, and this is not even taking into account that proteins are regulated in both space and time. So in space by confinements in subcellular structures, and in time throughout the cell cycle. So the central dogma in biochemistry, which was originally postulated by Francis Crick, states that the flow of information goes from DNA in the genome through a messenger mRNA, which is then translated into proteins that make up the proteome. And we now know that there's an increase in complexity associated with this process. So one transcript can be spliced in several ways. So this gives rise to approximately 100,000 transcripts from 20,000 protein coding genes. And these 100,000 transcripts give rise to an approximately equal number of proteins. However, the proteins can then in turn be post-translationally modified in different ways that further increase the complexity. And the proteome can also be organized in higher dimension structures, like proteins can engage in interactions, and these can be dependent on individual or combinations of PTMs. And this functional organization of it's this functional organization of the proteome that gives rise to molecular functions and phenotypes at the end of the day. And these phenotypes can then, for instance, be disease states that we are interested in studying. So the proteome and its functional organization through, for instance, the interactome is what gives rise to phenotypes such as disease states. So this is really the drug target sweet spot within omics. And today, many drugs and drug targets are both proteins. The method of choice for large-scale analysis of proteins and proteomes is mass spectrometry, and I will now give you the fundamentals of this technology. So a typical proteomic characterization follows this workflow. So it starts out with a biological sample. This can either be tissues, cells, biological fluids. Proteins are first extracted from this sample, and they are then digested into smaller peptides using specific proteases. And these peptides are then separated using an HPLC. And as peptides elute from this HPLC, they are analyzed by a mass spectrometer. And the mass spectrometer, it determines the masses of intact peptides and the masses of these, or fragment masses of these intact peptides. And this information can then be used to query proteome sequence databases that are typically generated from translated genomic information. This generates big matrices of protein abundance metrics across the analyzed conditions that can then at the end of the day be explored using different bioinformatic tools. And the basic principles of mass spectrometry, they involve a sample which is ionized, and these ions can then be analyzed in a mass spectrometer which operates at vacuum. And the mass spectrometer at the end of the day gives you mass spectra that can be functionally interpreted. Mass spectrometry is versatile, and you can analyze any type of ions, so not only peptides and proteins, but also metabolites, nucleic acids, and similar. For ionization, there are two main techniques. These are called electrospray and MALDI. And I also wanted to just highlight that a Nobel Prize in 2002 was given to these ionization techniques. So Koichi Tanaka developed the MALDI technique, whereas John B. Fenn developed the electrospray technique. So the mass spectrometer, which operates at vacuum, it has three main components. So first it has an ion source, which passes gas phase ions to an ion analyzer, and ions are at the end of the day detected using an ion detector. And the most commonly used mass analyzers are typically referred to as quadrupoles and ion traps, whereas the detectors are of time of flight or Orbitrap models. And as an example of how these instruments can look, I wanted to showcase the Exploris 480 mass spectrometer offered by Thermo Scientific. This system is based on electrospray ionization. It has both quadrupoles and ion traps for ion sorting, and uses the Orbitrap technology for ion detection. And here is a more in-depth view of the Exploris 480 mass spectrometer. And to indicate the flow of events, peptides are first ionized using this electrospray techniques. They are then analyzed and sorted using a quadrupole mass filter and a collision cell, which is an ion trap mass analyzer. Peptides are then shuttled through something called a C-trap into the Orbitrap mass detector, which gives you the mass spectra. So mass spectrometry-based proteomics can be used for both quantitative and qualitative analysis of proteins. And what I mean with qualitative analysis is protein identification, so just determining which proteins that are present in a sample. Whereas protein quantitation also involves determining the abundance of these different proteins. And these two are two independent processes in mass spectrometry. So when it comes to quantitation in a mass spectrometer, there are a couple of different approaches one can use. But I first wanted to highlight again the standard proteomic workflow, which involves cells and tissues from which proteins are extracted and digested into peptides. And these peptides are then analyzed by mass spectrometry, and informatic pipelines are used to interpret the proteomic data. So these are the different quantitative approaches that one can choose from. The two extremes are denoted metabolic labeling and label-free quantitation. And these color-coded boxes indicate where samples can be mixed in the proteomic workflow. So in metabolic labeling, cells are typically fed with isotopically heavy or light variants of amino acids that are incorporated into proteins. And this allows you to pool your samples already at the cell or organism level and thereafter do protein extraction and processing in parallel. So this, of course, reduces experimental variation associated with these steps along the way. The other extreme is called label-free quantitation. So this involves completely parallel processing of the conditions that are to be compared and independent mass spectrometry analysis of them. And the results are then compared at the level of data analysis. There are also some intermediate versions that are rely on chemical labeling or chemical derivatization of experimental conditions. And these chemical labels can be introduced either at the protein level or at the peptide level. The examples I will talk about in this lecture, they rely on chemical labeling at the peptide level using something denoted TMT or tandem mass tag approach, or completely label-free quantitation, which is the most versatile and commonly used quantitative approach in proteomics today. So just some brief background on first label-free quantitation. This is commonly referred to as LFQ. So it involves individual processing and analysis of the samples of interest, and the quantitation is simply based on comparison of intensities in the mass spectrometer. So it's comparison of intensities for the same peptide across your experimental conditions. So the second quantitative approach I will talk about is denoted TMT or tandem mass tag. And this is an approach that enables a combined analysis of multiple samples in one mass spectrometry run. And this is made possible through derivatization of peptides from different experimental conditions with tandem mass tags. These tandem mass tags are then released upon fragmentation, allowing quantitation of the different experimental conditions through specific reporter ions. So now that you have some background in basic mass spectrometry and its operations, I will go into applications and start out with the first example that concerns drug discovery and how this can be mediated through plasma proteomics. So using the latest and greatest workflows, it's possible to characterize plasma proteomes from humans in just a couple of hours. And this requires very little material, just a couple of microliters, whereafter plasma is isolated and the proteins are digested into peptides using robotic automated systems. The corresponding peptides are then analyzed using short chromatography gradients and fast scanning mass spectrometry methods. And at the end of the day, the mass spectrometry data can be analyzed and processed using automated pipelines that allows you to reveal proteins that are over or underrepresented across different disease states or conditions. And this allows you to identify so-called biomarkers, and biomarkers can be proteins that are associated with a disease state or disease progression or disease risk. This is illustrated here to the left, where this biomarker had very low levels in healthy controls, slightly elevated levels in a group with an increased disease risk, and even higher levels in the disease state. And monitoring such biomarkers in populations allows you to identify patients that would benefit from preventive measures or drug treatment. And the desired effect of this type of analysis is, of course, longitudinal monitoring of populations to allow you to intercept and reverse these disease states to maintain the population in a healthy state. So traditionally, biomarker discovery studies almost invariably involve this so-called triangular strategy. This first involves biomarker discovery and verification stages that can both rely on mass spectrometry. And these are followed by validation stages that almost invariably involve antibody-based or immunoassay-based validation of identified targets. The number of samples included in these different stages is indicated here. So typically the number increased by an order of magnitude for each step in this process. Meanwhile, the number of proteins decreased accordingly from thousands in the discovery phase to just a handful in the validation phase. So using the state-of-the-art mass spectrometry-based workflows for proteomics that involved automated sample preparation and processing, it's now possible to apply a rectangular strategy. And this rectangular strategy, it involves global analysis of all proteins in large cohorts for both the discovery and the validation phase. So it involves analysis of thousands of proteins in large cohorts of patients. And biomarkers can then be identified as or categorized as proteins that display differential abundance across different sample cohorts. So in summary, in this first example of mass spectrometry-based plasma proteomics, I highlighted how this technique can be used in biomarker studies. And these biomarkers are valuable for both disease diagnostics, but also disease progression monitoring. And these biomarkers can also represent drug targets. So this second example, it concerns mass spectrometry-based drug characterization and specifically focusing on assessing drug-protein interaction. And this second example, it concerns modulation of histone acetylation. And acetylation is a post-translational modification, and these acetylations on histone protein specifically are key epigenetic marks. So they are part of this histone codes that represents an additional regulatory layer of gene expression above the gene level itself. And these acetylations, they determine the chromatin compaction state, so how accessible the DNA is for RNA polymerase II and transcription factors, which is illustrated here to the right. And they are enzymatically regulated, so they are introduced by histone acetyltransferases or HAT enzymes, and they can be removed by histone deacetyltransferases or HDAC enzyme. So this is a dynamic modification system that enables cells to turn on and off different genes. These HDAC enzymes, they are common drug targets, and several inhibitors have been developed to modulate their activity. And regarding these HDAC inhibitors, so down here I first shown the chemical structure of acetylated lysine, and the first HDAC inhibitor is called SAHA or vorinostat, which is used as a therapeutic agent for T-cell lymphomas. And you see structurally it has a cap, a linker, and a functional domain. And this inhibitor, it structurally resembles the acetyl lysine and fits into the active site pocket of some of these HDAC proteins. But more recently, several new HDAC inhibitors have been developed that are now being used for therapy of different cancer types. But their specificity has been debated, and the example I will talk about concerns determining the specificity of this panobinostat, which is used for multiple myelomas. And the experimental setup is actually quite simple. It's based on what is denoted as a thermal shift assay. And the idea here is based on that proteins tend to unfold and aggregate with temperature, but ligands or drugs that bind to the proteins tend to have a stabilizing effect, which results in this shift in melting curve, which is then dependent on specific ligand binding to specific proteins. So this thermal shift approach, it can be multiplexed using the following setup. So starting out by preparing your cells and treating them with a drug. Thereafter exposing the system to a concentration range and extracting the soluble proteins only. These proteins can then be derivatized with these tandem mass tags, whereafter the samples are pooled and analyzed in one shot in a mass spectrometer. And the intensities of the reporter ions in this TMT system will then reflect how much of the peptide that is remains soluble at the corresponding temperature. And this allows you to generate melting curves for proteins and do this on a global scale in the presence and absence of different molecules such as drugs. Another way to do this experiment is to use different drug concentrations, but a single temperature. And this allows a slightly different readout, where the kind of remaining soluble proteins can be described as a function of drug concentration. And you can also do these experiments in multiple dimensions, which is illustrated in this heat map, which shows the differential stabilizing effect of a drug with respect to both temperature and concentration. So in the sample with panabinostat, blood was extracted from humans, and the blood was then aliquoted and exposed to a concentration range of the HDAC inhibitor. These samples were then exposed to a temperature gradient, where after PBMCs, so peripheral blood mononuclear cells, were extracted, and the solubility of proteins was assessed using mass spectrometry. So this analysis revealed 5 outliers that were stabilized using the HDAC inhibitor panabinostat. So 3 HDAC enzymes called with their systematic names HDAC1, 2, and 6 were all stabilized with the inhibitor. But also 2 other proteins like this zinc finger protein ZNF512 and the TTC38 were also stabilized. So this analysis then strongly indicates that panabinostat binds to and stabilizes these 3 HDAC targets, but that it also binds to or has stabilizing effect to these 2 proteins. And these may then either be unwanted off-targets of the inhibitor or desired additional targets of the inhibitor. So in summary, in this section where I talked about mass spectrometry in relation to drug characterization, I first talked about the thermal shift assay, which is an experimental approach that allows you to assess protein ligand or protein drug interactions that often modulate the stability of proteins. And using mass spectrometry, it's possible to do this on a proteome on a global scale, so you can do global screens of protein drug interactions. And this allows you to characterize the specificity of drugs, but also to repurpose drugs by identifying additional targets. So in this last example, I will talk about the drug repurposing and how this can be done using quantitative interaction proteomic approaches. First, regarding protein interactions, so protein they tend to engage in complex networks. This is illustrated here by protein interaction data from the simple eukaryotic organism budding yeast or Saccharomyces cerevisiae. And these interactome networks, they are generated using different approaches. So the red one is generated using a so-called yeast 2 hybrid approach, which is a genetic reporter system. The green interaction network is generated using affinity enrichment mass spectrometry, and the blue interaction network is generated using text mining, so global or universal querying of PubMed articles and abstracts for co-mentioned proteins. And this is interaction from a drug development perspective, as drugs frequently perturb and disrupt other protein interactions. And this is something one can utilize in drug repurposing. And the drug repurposing example I will talk about relies on interaction proteomics in a human cell. And I will first give you some background on how this is done experimentally. So the protein of interest, which is referred to as the bait protein, is fused to an affinity tag in a plasmid, and this plasmid is then transfected into a human cell that allows ectopic overexpression of your bait protein fused to an affinity tag. And after cell lysis, your bait protein can be fished out using antibodies towards this affinity tag, and proteins that co-purify with the bait can be quantified using mass spectrometry. And by doing this with multiple baits, one can generate interaction matrices where different proteins can be categorized as weak or strong or non-interactants with specific baits. And this allows you to build protein-protein interaction networks, which is shown in a small scale up here, or in a large global scale down here. And the example I will talk about involves drug repurposing related to COVID-19 and the SARS-CoV-2 virus. In this analysis, 27 coronavirus proteins were fused to an affinity tag on a gene level. And these constructs were thereafter overexpressed in a human cell line, where after the coronavirus proteins were affinity purified using specific antibodies towards the tag. And this allowed mass spectrometry-based identification, quantitation, and scoring of human proteins, and uncovering how these coronavirus proteins interact and interfere with human protein-protein interactions. And overlaying this new SARS-CoV-2 human protein-protein interaction map with information on available drugs and which protein interactions they target actually enables the identification of drugs, of already established and approved drugs that has the potential to target and disrupt interactions between coronavirus proteins and human proteins. So to sum up this part on drug repurposing, I talked about how proteins often engage in interaction networks, and that protein-protein interactions are frequent drug targets. And these protein-protein interactions, they are preferably studied by mass spectrometry-based methods in large-scale interaction studies. And this in turn enables drug repurposing. So to sum up this lecture, I first talked about proteins, and that proteins are both frequent drug targets and drugs per se. I also showed you and made a case for that proteins are preferably and globally best assessed by mass spectrometry. And I showed you how mass spectrometry-based methods can be used in drug discovery using plasma proteomics as an example, and how they can be used in drug characterization using this thermal shift and drug-protein interactions as an example. And finally, how mass spectrometry can be used in drug repurposing through analysis and uncovering new protein-protein interaction networks.